Welcome to week 4 of Pharmacology for Vocational Nursing 1. This week we'll focus on chapter 8. This is a very brief chapter in the textbook, but it's loaded with information. You're entering an exciting time in healthcare and genetics, and pharmacogenetics is expanding. You've heard me talk about pharmacogenetics, but what is this? Pharmacogenomics? What's the difference? Pharmacogenetics is the study of variation in drug metabolism and response. It focuses on how genes influence an individual's response or lack of response to a drug, whereas pharmacogenomics is the more global study of all the different genes that determine a drug's behavior. As we go through this lecture today, we're going to review basic principles of genetic inheritance, including nucleic acids, chromosomes, genes, and alleles. So let's get started. You'll remember that genetics and genomics, like pharmacogenetics and pharmacogenomics, are often used interchangeably. However, there is a difference. Genetics is geared towards the mechanisms of heredity and variation of traits, whereas genomics, like pharmacogenomics, is more broad. In some ways, it's more specific in that it is focused on function of the DNA. So let's consider why it matters to us as we think about pharmacology. There are diseases that are inherited. Some are present at birth, and we call this congenital. An inherited disease may be obvious at birth, or it may not. Genetic disorders, they're more commonly called disorders than diseases, occur when we have an error in the DNA coding that is inherited from a parent or parents, such as the case with the sickle cell disease, right? Sickle cell disorder. Or it can be a result of a mutation or a change, for example, in Down syndrome, where there's an abnormal cell division that results in extra genetic material from the 21st chromosome. So you see, some people inherit a genetic disorder while others are acquired through changes or mutations. A genetic predisposition is when we have genetic characteristics that influence our phenotypic development when there is an environmental influence present. In the absence of those influences, the disorder does not develop. Now let's take a quick brief overview. The field of genetics is quite expansive and it's a science all its own. But you may recall from high school science that DNA is deoxyribonucleic acid. It is a nucleic acid and it contains the genetic information and the instructions for structure and function in living organisms. Let's look at how these things all fit together. If we look at the structure here, you remember that the human cell, in every human cell, with the exception of mature red blood cells, there's a complete set of DNA molecules. DNA is that long sequence of nucleotides or bases. They're represented by ATCG. The order of the bases is what gives the exact instructions to the cell for its particular function. So our DNA are these two linear strands that run opposite to each other and then they twist together. This is called a double helix structure. Each of the DNA strands in a long linear molecule is made up of smaller nucleotides, and you recognize the nucleotides as adenine, cytosine, guanine, and thymine. When the two strands meet up, adenine pairs up with thymine and guanine with cytosine. So the genome is the total sum of the DNA in a human cell. This is a complete set of inheritance for an individual. Each person's genome is unique, the exception obviously being monozygotic twins or triplets. And the DNA of the monozygotic twins is identical, however, the expression of the genetic material is unique. Now, chromosomes are the organized structure of the DNA and the proteins in the double helix. Chromatin is the package, so to speak. So chromosomes are the pieces of the DNA. By packaging these chromosomes into the chromatin, it is smaller and it fits into the nucleus. This chromatin packaging also helps protect the DNA's structure and sequencing. The cell nucleus contains this DNA that's tightly wound up into the chromatin, and if we stretched it out, it would be about 6 feet long. In this compact package, remember, there are 23 pairs, 46 total chromosomes, and that the first 22 pairs of those chromosomes are autosomes. They're alike in both male and female. That 23rd pair are the sex chromosomes, and they determine gender. A female has 2 X chromosomes, one copy inherited from each parent, and the male has an X chromosome inherited from his mother and a Y chromosome inherited from his father. So let's delve a minute into the Human Genome Project. In 1986, the United States Department of Energy began the Human Genome Initiative. You're probably asking yourself, what is she talking about? What about the Department of Energy? Well, the Department of Energy was the department created from two agencies, the Atomic Energy Commission and the Energy and Research Development Institute. So following the development and use of the atomic bomb, the Department of Energy was charged with the task of researching the effects of the atomic bomb and radiation on humans. This was most easily done by looking at the cellular level and the DNA. You probably remember, or maybe you don't, that we have known about DNA since 1869. It was first correctly modeled by Watson and Crick in 1953, almost 100 years later. So that's why the Department of Energy started the project, but they quickly realized that they needed help. So they teamed up with the National Academy of Sciences, who then passed that to the National Institute of Health. The National Institute of Health was concerned with the health and wellness issues of the project, whereas the Department of Energy was concerned with the social implications of the use of atomic bombs and radiation. It quickly became an international project. Now, the Human Genome Project was completed in 2003. Earlier than so, just a little note, did you notice that 2003, 50 years from 1953, the celebration? Anyway, this continues to be the germinal research behind continued improvement in the prevention, treatment, and finding cures for diseases. It also helps in the development of new tools for genetic data analysis and storing genetic materials. So, of course, as you can deduce, genetics involves not just the science of genetics and genomics, but also ethics. There are legal and ethical implications of human genome research. And we as a society also must balance this with personal choice and a person's right to privacy. So let's see if we can make this a little bit more real. We've discussed pharmacogenetics, genomics, and aside from drugs, there's also gene therapy. Gene therapy is different in that it's the transplantation of a normal gene into the cell. It can replace a mutated gene that causes disease and thus create a healthy copy of the gene. It can inactivate or knock out a mutated gene that's not functioning properly, or a new gene can be introduced into the body to help fight a disease. Unfortunately, it's not quite as simple as just inserting the gene into the cell. It usually doesn't function, so a carrier is genetically engineered to deliver this disease. You have a simplified visual in your textbook showing in vitro manipulation. This is what we're talking about. I have included here a little bit more detailed model to show you that some viruses are modified so that they cannot cause disease, but then they're used as vectors because what they do is they deliver that new gene into the cell by infecting it. They integrate their genetic material, including that new gene, into the chromosome of the cell. We have been using recombinant forms of drugs for decades. Recombinant means that two molecules of the DNA exchange pieces of genetic material. For example, after recombinant bacteria was grown on an industrial scale and scientists learned how to harvest the insulin they produce, Eli Lilly, a drug company you may recognize, brought Humulin into the market. This was back in 1982. Scientists have since used recombinant DNA technology to produce other products like human growth hormone, vaccines, interferon, medications you'll learn about later in the year, epigen, interleukin, activase, maybe you've heard of clot busters, or parathyroid hormones for TAO. So you can see why regulatory and ethical issues have risen from this science. Now, eugenics is the science of improving the human population or humans themselves by controlling reproduction to increase desirable heritable characteristics and traits. The ability to alter genetics prior to birth or early creates fear and comes with many questions we cannot possibly cover in this course. But be mindful of the controversies and remember that it is the patient who is at the center of all that we do as nurses. Earlier, we discussed chromosomes and chromatin, and you may recall that I used the term allele. Alleles are multiple versions of a gene. Each person typically inherits two alleles of each gene, one from each parent. Polymorphism means many forms. So the variations that occur with DNA, along with the environmental and the dietary factors, create our susceptibility to disease or our response to treatment. The concepts of alleles is important to pharmacology because many of the polymorphisms, or the forms that change the various protein production or function, are evident when we look at the effects of what we give in terms of medication. So what does that all mean? Let me see if I can simplify it for you a bit with an example. There are two patients. One has a genetic sequence, or alleles, AAGCTA. Another, AAGTTA. Notice the only difference between these two is that there is a polymorphism in the substitution of the thymine here for the cytosine. So these are two alleles, or two versions of the gene. Now you're learning about cytochrome P450. There are many genes responsible for drug metabolism and the activity that the different alleles have on these systems. So many of the genes responsible for drug metabolism and activity have different alleles on the same gene, thus producing different metabolic effects. This is why we have poor metabolizers, intermediate metabolizers, extensive metabolizers, and ultrametabolizers. Poor metabolizers may lack the ability to make a functional enzyme. Intermediates may carry a defective gene. Extensive metabolizers usually carry two functional alleles, whereas ultrametabolizers may carry multiple copies of the gene, thus producing more enzyme than a single or even two functional alleles could do. This leads to the development of personalized medicine. So that's where we tailor medication and treatments to individual characteristics of patients. Researchers and specialists in medicine are using DNA microarray technology to examine and monitor gene expression and detect polymorphisms and mutations, very much like we would look at tissues from biopsies. Clinically, there are many implications and applications of pharmacogenomics, and we could see that there are as many as there are people if we consider that each person may someday have individualized medicine. Step aside, Spock. Bones is here, and he says, "Damn it, Jim, I'm a doctor, not a computerized robotic microscope. Now, where did I put my tricorder?" Just like Bones, though, as we study and practice in the clinical setting, let's remember that we're dealing with people. And don't forget our nursing implications for what we need to do as nurses. Always take a thorough patient, family, and drug history. Recognize there are situations that may warrant further investigation through genetic testing. Help identify resources for our patients. Always maintain confidentiality and privacy. It's really important with genetics, especially because there are so many controversial issues surrounding this area. Ensure that informed consent is obtained, although we'll talk more about that. Informed consent is not your responsibility to explain those genetics or those tests. It is your responsibility to make sure that the patient has been informed. And then, of course, we need to recognize that patients may need genetic counseling, and helping them to be able to identify resources will be valuable in your nursing career. As always, if you have questions, please don't hesitate to give me a call, send me an email, text, or post in the discussion forum. I look forward to our discussion this week, and till then, take care.